Good morning, afternoon, evening for all of you, wherever you are. My name is Shuchi Datta, and I am going to be your tour guide. Before starting, I wanted to point out that on the RCSB website, so if you go to the RCSB website and under documentation, there is extensive documentation and examples and FAQs, etc., that are written related to MolStar. And so I invite you to check this out if you haven't already done so. And I also wanted to point out that we welcome questions from you related to the specific tools and features that we offer. So if you have things that you're looking for and can't find, or if you have requests for specific examples, please reach out to us. Use the Contact Us button over here to reach us, and we will try to answer your question. And if it is something that others can benefit, we will add it to our documentation. So with that, I'm going to begin today's tutorial or webinar and get into slideshow mode. So as I was preparing the agenda for this webinar, I was looking for a story, something that we could use to showcase the MolStar features and functions that we have. And I came across this announcement from the FDA about the first gene therapies to treat sickle cell disease. And I figured sickle cell is something that has a lot of firsts associated with it. So I thought this would be a good example for us to use for this webinar. So I will begin by giving you an introduction to the storyline so that there is some meaning to all the clicks and functions that we explore. And then I will tell you a little bit about the tool, and then we look at the actual structures. So to get started, I will take you back about 120 years. So in 1904, Walter Clement Noel traveled from Barbados to Chicago to study dentistry. He had what was not known at that time, but he had various sores on his feet. He would get frequent infections. He would have these episodes of not being able to breathe, etc. And in one such episode, when he visited a doctor in Chicago, they did some routine tests. And one of the things that they did was a peripheral blood smear on a slide. And they noticed something unusual in his blood. And this was eventually reported by James Herrick in 1910 as the first reported case of sickle cell disease in our current literature. Of course, later it was understood, or it was known, that this kind of a symptom and phenotype had been seen many, many years ago in Africa and in other places. But this was the first report of sickle cell disease. And then several years later, James Van Niel was able to figure out the inheritance pattern of sickle cell disease. Linus Pauling was able to show that this is a molecular disease, and that sickle cell trait and normal hemoglobin and sickle cell hemoglobin, they actually could be separated, and you could see these different peaks showing the different populations of some molecule, hemoglobin. And then it was around that time that Max Perutz was working on the structure of hemoglobin, and we were able to understand what hemoglobin looks like and understand the difference between different species hemoglobin, oxy, deoxy. And as we see here, we began to understand how oxy and deoxy hemoglobin undergo conformational changes. And by 1980s, we had a structure of sickle cell hemoglobin with the mutant form of sickle cell, mutant form of hemoglobin, showing how that changes the behavior of hemoglobin. And so sickle cell disease, even though it is a single amino acid mutation, it can lead to pretty serious consequences, some of which are listed here. I'm going to draw your attention to vaso-occlusive crisis or vaso-occlusive episodes, VOE or VOC, as these severe pain episodes that individuals who have sickle cell disease experience. And this, I'm mentioning this particularly because monitoring how frequently these episodes occur became sort of the metric for measuring how successful the treatment options that were being developed are. So that's why I thought I'd mention it here. So what happens in sickle cell disease is that mutant hemoglobin's valine in the deoxy state, it is a hydrophobic side chain that sticks out and it buries its head in the neighboring hemoglobin and so on and so forth to form these long polymers or fibers, which then inside the red blood cell cause the blood cell to sort of distort. And this distorted or sickle cell shaped red blood cells, they kind of get stuck in small capillaries, leading to blockage and lack of oxygen supply, leading to these various symptoms. And so this is what is happening. So that one single mutation can cause all this, leading to organ damage, loss of vision, stroke, et cetera, et cetera. So continuing the timeline, in the 1990s, it was noted there were certain people who had a very mild form of sickle cell disease. And this was associated with the presence of this hereditary persistence of fetal hemoglobin. And this actually inspired research and treatment leading to the use of hydroxyurea to treat sickle cell disease. So hydroxyurea actually promotes the production of fetal hemoglobin, and that somehow balances out the sickle cell and the fetal hemoglobin's proportion in the blood, allowing the individual to have a reasonably normal life and not have to experience all this pain and organ damage, etc. In the 2000s and 2010, more research, actually more specific research, identified this particular transcription factor, which is a repressor of the gamma globulin, that if present, it transitions or it allows the transition of human babies from the fetal hemoglobin to the adult hemoglobin. But in the absence of this, the fetal hemoglobin or the gamma hemoglobin is it continues to be produced. And that has been described as one of the scenarios in which the individuals with sickle cell disease mutation can have a relatively mild form of the disease. And so this idea was used to actually target a therapeutic approach. And so I'm going to quickly summarize, and that is really what the one of the gene therapy approaches uses. So to quickly summarize, some of the currently used or currently available FDA-approved treatment options include hydroxyurea, which increases the production of fetal hemoglobin, voxelotor, which we will look at, that actually it's a small molecule drug that favors the oxygenated version of the sickle cell hemoglobin formation, and therefore the sickling effect is reduced. Crizanlizumab, this is something that we will not talk about today because this acts not on hemoglobin itself, but it interferes with the ability of these sickled hemoglobins to get stuck in blood vessels. And then the two gene therapy approaches, Lyfgenia and Casgevy, these are the two approaches that we will work towards understanding. Now, I'm going to pause here and see if there are any questions in the chat or any comments. And I don't see anything. So I'm going to continue. So the next part of the agenda was to introduce you to the tool. So of course, in the third part, when we are doing this demo hands-on activity, we will get a chance to explore all the tools and buttons and options. But I just wanted to point out that the MolStar that we are looking at over here is the MolStar version that is associated with the rcsb.org website. So you don't need to install or download or assemble anything. As long as you have a modern browser and you are able to get to the rcsb.org website, you can access this page or this tool. And there are two ways in which you can use the tool. One is where you're looking at structures starting from the structure summary page. So every single structure in the PDB has a structure summary page, and linked to that is the MolStar tool. But you can also use the MolStar tool, a standalone version of it, to visualize structures that are not on the rcsb.org. So these may be structures on your local disk or files from another resource, such as AlphaFold or some other source, as long as you have the complete URL path. And or if you have it on your local disk, as long as you can upload it, you can visualize these structures on this rcsb.org's standalone MolStar version. Okay, so now the things that you can do. Oh, and I wanted to point out that when you open the MolStar tool, you will see three main sections. The top left, this section is the sequence panel. This is where all the polymer chains and any small molecules or sugars or whatnot that are present in the structure, you will see a listing of their components, their sequence. This is the 3D canvas, this white space, and we will look at it in detail shortly. This is where the molecule shows up, and you can rotate it and zoom in and measure things and compare things, etc. And this is your control panel. This is where you select the various options and give instructions for what you want to see in the 3D canvas. And this will become more obvious as we go along. So I wanted to point out upfront, and we will sort of review this at the very end, the molstar.org tool not only allows you to visualize structures and look at whatever coordinates are available that you have uploaded. So you can see the whole structure, you can change representations, you can color them, you can view experimental data if available, symmetry, you can look at it, you can measure angles, lengths, dihedrals, etc. You can save images and animations. But on top of that, there are ways in which we have created opportunities for you to map annotations from a variety of bioinformatics resources like UniProt, ScopCath, etc., and see where the specific annotations, like let's say the active site or the binding site or a specific domain, where do those annotations map on the structure? We'll look at that provision. We will show you options for comparing structures and then visualizing that in 3D using the MolStar tool. And finally, this option is to look at groups of structures and to look at patterns, trends, and things like that. And this will also come in handy that we will look at in one of our examples. So I see there is a question answer thing. Okay, so the question is about standalone MolStar. No, we don't need any additional software to be downloaded, and I'll show that in a moment, how we can access the standalone MolStar. So, and I see a question about compare structures. So up to 10 structures can be superimposed on the target that you choose. And we will, if you hold this thought, we will get to it shortly. So with that, I'm going to start our visualizing molecular stories journey. And to help you have access to these PDB IDs, I've actually created a companion document, and I'm going to share that link here in chat. So I invite you to open this, and then you can save it in your local area and then edit it as needed. While you're doing that, there's a question that asks me, is MolStar for proteins alone? Yes, you can display RNA. We will, towards the end, look at an example that has not RNA, but DNA along with a protein structure. You can also look at carbohydrates, any ligand, etc. So this is for all kinds of biological molecules. Okay, so I'm hoping that people were able to look at this document, and you're welcome to follow along. And so what I'm going to do here is we will begin by exploring hemoglobin structures. So we want to understand sickle cell disease and the gene therapy for sickle cell disease. But I think it is probably a good idea to start with hemoglobin structures. And here, let me share this again. Okay, so hemoglobin structures, as we saw in an animation earlier, so we are going to look at both oxyhemoglobin and deoxyhemoglobin, and use this opportunity while we are exploring hemoglobin structures to learn a few tips and tools related to MolStar. And so let's get started. So this is where we are headed. Let's go to the PDB. And if you are following along with along in the document, so we are going to this PDB entry 2DN2. This is deoxyhemoglobin. So you can start by putting in the PDB ID on the top search bar, and you come to 2DN2. And over here, you get a quick glimpse of what this molecule looks like. And up top here on the structure tab, if you click there, or if you click on the structure hyperlink here, you can open the MolStar tool with this particular structure loaded. So here we are looking at 2DN2 in MolStar. And we see these 4 chains representing the 4 polymers that make up this structure, the hemoglobin, the 2 alpha and the 2 beta chains. And what we are also going to see here is our sequence panel. And in the sequence panel, we have sequences of the alpha chain, beta chain. This is the heme, water. So this is deoxy. There is no oxygen associated with it. And in here, if you look at the controls panel. So the controls panel shows you the various polymers, the ligands, the waters. I can hide or show any of these components that are currently being displayed. And if I choose, I can actually change that representation. So for example, in the polymers, I can go and say, I want to see this in the molecular surface view. And so notice how it was able to create the molecular surface for this. And I can then say, okay, this is great, but I want to, so I did it twice. I'm going to delete this one. So it was as simple, you can delete it or just simply hide it. Now what I want to do is this molecular surface currently shows 4 chains in 4 different colors, but I don't know which ones are alpha and which ones are beta. So if I want to change this color representation, instead of it being colored by chain ID, I can change that and say, I want to color it by entity ID. So the alphas have one entity and betas have another entity. And as I mouse over this, you see it's saying hemoglobin beta subunit at the bottom corner, and the green chains are the hemoglobin alpha subunit. Now, this is, I know it is symmetric or somewhat symmetric, but here is an opportunity for me to explore the symmetry of this molecule. If I click and enable the symmetry option, here I can see that these 2 alpha chains are related by 2-fold and 2 beta chains are related by 2-fold. And then it's a sort of not typical, not strict 4-fold symmetry. Okay, so I am looking at questions in the from you.
Suchi, I sent you everyone's questions directly, so you don't have to scroll through the chat. You're welcome.
Yeah, thank you so much. So I am going to pause here and take a few questions. So the question that I have is, so let me hide this. So I'm going to hide the surface view for now. And I have questions that say, how can I hide or select sequence or show part of a structure? I'm going to come to that in a moment, or maybe I can do that right now. So actually, hold that thought for a moment. How do I highlight a particular amino acid? Okay, it's all about amino acid highlighting. And so that's, in fact, the next thing that we are trying to do in our steps described in the component. Sorry, this is getting a little confusing. Okay, so we are back in our view here. So what I wanted to repeat, and so I'm going to open another window. And in this, I'm going to look at the oxyhemoglobin. And so the oxyhemoglobin's PDB ID is 2DN1. This is the oxy form. And I'm going to repeat the steps where I'm going to look at this structure. Now, I wanted to draw your attention to this fact that here, when I uploaded the structure, it came up with 2 different colored chains. It did not show the very typical 4 chains that we had seen in the previous structure. And that is because this particular structure was deposited as 1 alpha, 1 beta chains, and then the symmetry related alpha and beta chains is what makes up this assembly, this tetrameric structure. And so this is something you can explore by changing so this is the submitted coordinates, the model, and what we were looking at was the assembly that we have generated for you so that you can look at the tetrameric structure. And the reason I point this out is that depending on the structure you are exploring, you may have to choose between whether you want to see the submitted coordinates or the model or the assembly, depending on what you are trying to explore and visualize. And so here, again, if we generate the assembly, the symmetry, and now display the surface, what I want you to pay attention to here is that in this structure, the space in the center is very, very minimal compared to the other structure, where, if we put back that, there was a lot of space. And so this is really a very easy way to demonstrate to your students or for your own exploration purposes that so this is referred to as the taut, and this is referred to as the relaxed version of the hemoglobin. So the relaxed is all open and loose, and it's touching each other, whereas the taut version is all tight and clenched up. And so this is different, and this is the distinction that you see in deoxy versus oxy. And okay. Having said that, as we continue through this, one of the questions that asks in the companion sheet is that, can we examine the neighborhood of one specific amino acid? And this is something that, if we go to the, and so I'm going to do this in the deoxy. This is the structure where I need to do this. I'm going to hide the surface. And I'm going to pay attention to the sequence panel here. So in the sequence panel, I have the alpha chain, and this arginine 141 is what the question is all about. So if I click on this, so let me reload this just so that there is no confusion. So I'm going to click on this arginine 141. Now, this is where I wanted to point out. So if you noticed, the moment I clicked on it, it jumped onto this arginine 141, and it's showing you all these various interactions that are stabilizing this. So MolStar has two modes that you want to be aware of. This is the default mode. So in the default mode, if you click on anything in the structure or anything in the sequence panel, the instruction that it gets is that I need to quickly center on that particular residue or ligand or whatever you have chosen and display the neighborhood and show all the non-covalent interactions. So when I wanted to examine the neighborhood of arginine 141 in chain A, that's what I did. And it is showing me. And so now I can mouse over each of these amino acids to see what they are. So that's aspartate 126 in chain C and tyrosine in chain, another chain, and so on and so forth. So this is great if you are interested in just one amino acid. But what happens if you want to select several amino acids, or you want to not just jump and look at the environment, but I want to, let's say, change the color. I want to do something else with this. So in order to do that, I need to activate the selection mode. So I click on this. And now notice a new panel appear. In this state, which is the selection state or the selection mode, if I click on all this and zoom back, notice that whole stretch gets highlighted, okay? And as it is highlighted, it is colored in a green color, or it's highlighted both in the sequence panel and simultaneously in the 3D canvas. Once this is selected, I can say, oh, I want to change the color of this to be black, okay? Right? I can take this region and say, oh, I want to take this region and change the representation of this to be, let's say, space fill. And what this is going to do, it is that it's going to create a new component over here. And so that I'm able to identify what component this is, I can give it a name and create this component. So now this test component that I created in spacefill mode is displayed. So I hope this addresses some of the questions that had come up earlier about, can you select a stretch of amino acids or any particular feature and then change representation or change color or things like that. Okay. And the question, so how do we activate selection mode? So this toggle switch turns on and off the selection mode. If I click on anything now, so because there is no horizontal menu over here, if I click on anything now, it's going to jump in and show me that particular residue. But if I have this on, I can select anything on the 3D canvas or also on here, and it'll show up. I hope that helped. Okay, so I'm going to go back to our, so I just reloaded this, and I'm going to go back to that arginine 141, because one question that it asks in the companion worksheet is, what is the distance between this arginine's side chain and the closest side chain of an opposite charge? So that is this aspartate 126. So basically, I'm trying to measure this distance, okay? And so in order to measure a distance, you have to specify two endpoints. So if for an angle, you have to specify 3 points and so on. So I'm going to activate the selection mode. And because I'm going to measure atom to atom, the level of selection, if I keep it residue, if I click on this, it selects the whole residue, but I only want to select an atom. So I change that level to atom. And now if I click on this atom and that atom, this is the distance that I want to measure. So I come into this side and under measurements, say add. And notice here, the selected atoms are already listed. So if I have only one atom selected, all I can do is to label it. If I have two atoms selected, I can measure distances. And so I'm going to do that. I click on this, and it's telling me that the distance is 2.78. It's written here and also on the screen. Okay, so I see a question about making surfaces. We will do that again for another structure. So hold that thought, and we'll get back to it. But I hope you saw how you can identify 2 points, any 2 points in the structure, and you can measure the distance like this. And if I had 3 atoms selected, I could measure an angle, dihedral, for with 4 atoms selected, so on. So what we have done so far is we have learned how to visualize structures. We have learned how to display things in surface mode. We have changed colors. We have changed representations. We have examined the neighborhood of a particular amino acid, or it could be a ligand or anything. And we've learned how to measure distances. So let's go back to the slides. And just as a quick reminder, we saw the difference between oxy and deoxy hemoglobin. And we were able to create a view like this and measure distances, right? Now, what if you are interested in specific ligands? Let's say you're looking through the PDB and you're looking at structures with a particular drug bound to its target, protein or nucleic acid, whatever the target may be. So what we're going to do is we're going to look at hemoglobin. Think about hemoglobin as the drug, as the ligand, sorry, not as the drug, as the ligand. And I'm going to show you that if you come to the structure summary page, so this time we are looking at the oxygenated form of hemoglobin. If I scroll down, so here's a lot of information about the macromolecules, the alpha subunit, the beta subunit. And if I scroll down, here is information in the small molecules section about the various ligands. And I am going to look at where the oxygen is bound. So my question is, what does the oxygen interact with? And so to do that, if I click on this interactions option, there are 2 flavors that I could choose from, the chain A oxygen binding and chain B. So we'll do both one at a time. So in chain A, if I click on that, it opens MolStar and it centers around oxygen and shows you what is going on in the neighborhood. So here we see that the oxygen is bound to the heme. Here's your proximal and distal histidine. And as you mouse over it, you can see the identity of the histidine listed at the bottom. Okay, so this one is histidine 87. And if I go back and repeat that for chain B, okay. Here, the proximal one is histidine 92. So these are 2 different polymer chains, 2 different proteins. They have different sequences, but they have the same sort of organization of the histidines holding the heme and the oxygen binding to the heme. So that's essentially what is where oxygen binds, right? And so a question that comes up in the chat here is about, is this residue number the residue number of the full protein or the whatever is in the structure here? And this will actually, so you might encounter this, that the uniprot numbering, which represents the entire protein's numbering, compared to what is in the PDB structure, they don't always agree. And so if there are differences, both the numbering are denoted one within a parenthesis. And so it'll as we go through this, you might see examples. I hope that answered your question. So what we wanted to do here is we wanted to okay, look at where these, the oxygen is binding. Oh, and I did want to show you that wherever experimental data is available, so if we go back, the other tab, sorry. In this, if we, sorry, scroll down to that small molecule section, notice there is an interactions and density. So this, wherever experimental data is available, if I click on this, it will not only open the MolStar showing you that particular ligand, but it will also show you the electron density around it. And notice all this got activated because we have the structure factor and information here. We can display the electron density and you can explore and examine it. Of course, you don't need to come to the electron density just this way. You can upload the structure in here and then also just activate the density and visualize it. So while we are doing this, I wanted to take a moment and answer the question about standalone MolStar. So standalone MolStar is available from this top menu up here, okay? So standalone MolStar, I can activate. I'm going to open it in a new tab. So standalone MolStar comes with nothing loaded to it. You can open files, you can select a file from your local drive. You can also download structures from the PDB. You can type in the PDB IDs, or if you're looking at computer structure models, so the ones that are available from rcsb.org, if you put in the full identifier, you can upload it this way. But I also wanted to point out that in the MolStar tool, what we didn't talk about is here. Let's say we are doing this examination and we really like this view. We want to save this. So the way to save an image is you click on this camera option and you can download this image. Or you can also save the state, which is all of this. It saves the information about the orientation of the molecule, what is being shown, etc., or the whole session. So the value of using the session option is that you can save the session even if you have uploaded your own coordinates and you haven't, that file is not available in the RCSB. So the state is only functional if all the information is available, all the files are available from, let's say, rcsb.org. But here, everything will be saved. And once you have a session saved, you can share it with someone. And for example, I have, let's see if I can pull up. I think I have a session saved from before. And what I can do is I can just drag and dump it into this. And so for example, this session, so I'm just pulling it from my menu and dumping it here. And voila, this is a structure that I was examining at some point in time and I had saved it. And so it came up with all the various selections I had made, the colors I had chosen, things that were highlighted, etc. So the standalone MolStar tool can come in very handy for sharing and collaboration if you have something you make and you share it with your collaborator. And then, okay, I have a question here about, is there a way to show the hydrogens? Yes, there is a way to show hydrogens. And if the hydrogens are present in the structure by default, so let's just clean this up. If you come up top here, and under these additional information options, there is a hydrogen show all. So it's always in the show all default mode. But if you want to hide it, you can hide all. And so there is a way to do it. I have a question about differences between NGL viewer. There's actually a whole document that was produced. So I invite you to go to that, document section that describes, so NGL to MolStar migration guide. So please feel free to check that out. And hopefully that'll answer your questions. Okay, so we've done enough exploration of our oxy-deoxy hemoglobin, but we wanted to learn more about sickle cell hemoglobin. And so sickle cell hemoglobin, if we follow the same steps, and in the interest of time, I'm going to expedite this. So if we compare the deoxy and the carbon monoxy sort of is a mimic of the oxy form of the hemoglobin, sickle cell hemoglobin. You see, it has the same behavior of the taut and relaxed conformations. And but this still doesn't explain what is going on in the sickle cell disease that leads to the disease. And so let's look at that a little bit closely. So I'm going to go to the structure 2HBS. And in this, I am going to actually, so notice here, the biological assembly looks like the same old hemoglobin that we were looking at, but actually the deposited coordinate is more than that. And this is what I want to see in the structure. So I want to see the model. And here, if you look at this structure, there is interaction or close proximity between this. And as I'm mousing over, you see that this is the valine 6 of the beta chain. And if I click on it, this is sitting in this hydrophobic patch of this alanine, this phenylalanine, and this leucine. So hydrophobic side chain finds a home in this hydrophobic patch. And this is really the primary interaction that begins the polymerization, which will lead to the sickling and all these complications. So understanding this was very critical. And now I want to draw your attention to this. I'm going to activate the selection mode and show you this side chain, which is of significance. And I want you to keep that in mind. That is this particular threonine 87 sitting right next to that hydrophobic pocket, almost approaching the this beta chain, but not quite. And this is what is happening in sickle cell hemoglobin. Now, in our earlier storyline discussion, we learned that this particular sickle cell disease, by treating it with hydroxyurea, what happens in the individual is that the production of hemoglobins, the fetal hemoglobin, is increased. So if we looked at the fetal hemoglobin structures, again, this is deoxy and carbon monoxy, or oxy, versions of the fetal hemoglobin. Overall, it looks very similar. But what I do want to show you is that the polymer sequences are very different, and they have actually many distinct differences that allow the fetal hemoglobin to bind to oxygen more tightly, and that's advantageous for the fetus to be able to absorb oxygen from the mother. And this is something that we will look at. However, let's look at a fetal hemoglobin structure, 1-FDH. And I wanted you to note that as we scroll down, in the small molecule section, we saw how we can jump from this page to the MolStar. But I wanted to show you also the possibility of looking at other structures in the PDB that have the same chain. So the difference between adult and fetal hemoglobin is the presence of this gamma chain versus the beta chain in adult. And so this gamma chain, I wanted to see what other structures are there in the PDB with this gamma chain. And to do that, in this macromolecule section, I'm going to open the uniprot group. So all uniprot structures that have this uniprot ID, I'm going to open it up. And you can examine this in detail, but I'm more interested in the sequence, and actually the better view is the explore in 3D, where I'm seeing all the sequences of the gamma chain present in a structure. So the PDB IDs are listed here, and the structures are listed, shown here. I'm going to see if there are other structures in the PDB that have the gamma chain, meaning they are the fetal hemoglobin. And what I see here is a mutation. So if I click on this and zoom in, so right now I'm not seeing anything because I haven't activated this. So I want to see this structure, sorry. And I wanted you to appreciate how just by these few clicks, I'm looking at the structure of another fetal hemoglobin that has a mutation at this particular location. And how do I know the significance of this location? I can display the other polymer chains, and this is near heme. And long story short, this is actually a documented mutation, the Tom's River mutation, which is a valine 67 to methionine mutation that has problems with binding oxygen simply because the side chain is so much larger. It occupies that binding space, and this causes babies born with this mutation to turn blue. And the good news is that as the fetal hemoglobin level declines and the adult hemoglobin kicks in, if there is no mutation in the adult hemoglobin, the baby is fine and everything is good. So this was just an aside, but we go back to our fetal hemoglobin story, and this is so the question that we really want to ask is how similar are the sickle cell and the fetal hemoglobin structures? I mean, we are seeing this being used in therapy, but are they similar? Are they different? How do we figure this out? So under the analyze options in the top bar, if we click on this pairwise structure, as soon as it loads. Okay, so I'm going to align to HBS, which is the sickle cell hemoglobin that we were looking at, where the valine was interacting with that hydrophobic patch. And I'm going to look at 1-FDH. This was the fetal hemoglobin that we were looking at just a few moments ago. And now I'm going to compare not the alpha chain, but the beta chain in both these structures. Here, notice there was someone who had asked, are the chain IDs and residue numbers always consistent? So in this case, you may see changes in chain IDs, the author-defined chains, and the chain IDs that we have used for PDB's annotation curation purposes. If there are discrepancies, you will see them represented in this format. So we are comparing the hemoglobin beta chain and the fetal hemoglobin's gamma chain, the one with the sickle and sickle cell disease, and this is the one with just the fetal hemoglobin. And we click on the compare button. Before I do that, I want to point out that there are several different algorithms using which we can do the comparison. We are just going to use the default options for now. And if you have questions, there is, again, extensive documentation. If you click on that help page, it'll take you there, give you extensive documentation. So we've done the comparison. We see that the identity in structure is only 73%, but the root mean square deviation, so if you superpose the carbon alphas, they are really, really, really similar. And what I wanted you to pay attention to is what we are seeing here is just the one chain that we compared, but we would like to know where is the rest of the structure, right? So again, we use these boxes to show additional polymer chains and additional non-polymer components to be displayed here. I can center on this. So these structures are very similar. Just for your information, I can expand this view and look at the whole thing and do my measurements or coloring, changing representation, etc. Or come back. I wanted to draw your attention to that position 87 that we were looking at. Okay. And it is this particular position. So in the sickle cell hemoglobin, it is threonine. In the fetal hemoglobin, it is Q. And this idea of having glutamine at this position actually inspired the creation of this other gene therapy approach, where if you put glutamine in this position, and if you remember from the earlier view, where we were looking at how close this threonine was to this gray chain, if you put a glutamine here, this will actually push it out of the interaction space. And hence, this particular change or engineered mutation is actually called an anti-sickling mutation. And this is utilized in one of the gene therapy approaches that we will look at. Okay. So with this, we've explored several hemoglobin structures. In the remaining few minutes, I wanted to quickly point out that the CRISPR technology that is used in one of the gene therapy approaches actually doesn't touch the hemoglobin. It touches the gene repressor for a gene for this particular protein called BCL11A. This is a transcription factor that represses gamma production, the hemoglobin gamma production. So the CRISPR technology that is used in the gene therapy approach actually destroys the gene producing BCL11, which in turn then is unable to repress the gamma, and therefore fetal hemoglobin production is increased, leading to reasonable, ability to, breathe and perform activities even under low oxygen conditions. So in the interest of time, I'm going to skip the detailed analysis. So in here, there are steps that walk you through the various explorations, or we could do this very quickly. So the PDB ID that we are going to examine here is 6Ki6. Yes, 6Ki6. So let's go to 6Ki6, 6Ki6. This is your, it's actually a part of the full protein. And if we look at it quickly in the MolStar, you can see that there are several zinc fingers. So 1, 2, 3 zinc fingers. If I click on the zinc shown here, it shows you that each of these are 2 histidines and 2 cysteines binding to the zinc. Okay. And there was a question earlier about being able to see nucleic acids. So yes, nucleic acids are displayed. So here is nucleic acid. And if I click on any atom, it'll actually show me the full atomic view. I can do measurements, analysis, so on and so forth. What I wanted to show is that in this same structure, if I click on the sequence annotations over here, this is something that we haven't seen so far. This shows you a mapping of all kinds of annotations about this structure. For example, in here, we have these annotations about the ranges of the zinc fingers. So if I click on that, it'll focus on that whole zinc finger. If I go to the other one, it'll move to that zinc finger. Then there are, I can show only one helix. I can look at these other different annotations that are displayed on the sequence. It can simultaneously be seen in the structure over here. So this, I think, is a very handy way of exploring the structure. And then I wanted to quickly point out that this, if we come down to the explore option here, now we can see that here, this is the AlphaFold. This is the full BCL11A protein. What we were looking at is just this part. So in a lot of structures that are multi-domain, you might come across structures that are only one or two small parts or domains of the whole protein. But you can visualize the whole structure. So this is the AlphaFold prediction. And on top of that, I can display this particular structure that we were looking at a few minutes ago. Now, your value of doing a display like this is I can then say, okay, show me where is my polymer chains. So in this particular structure, we had DNA bound. So now we can see in this superposed fashion where the DNA would lie in this structure. But this also highlights the point that a lot of these AlphaFold structures have parts that are all this loopy and disordered and so on and so forth. And so when you are looking at these disordered parts, you have to, take it with that particular caution. And these large loopy parts, notice they are all colored in orange, while the parts that are more ordered are colored in blue. And this is where we have more information, more data, and so I wanted to showcase that just by clicking a few buttons, you can display multiple structures. I can display other structures also, and so on and so forth. Okay, so now to try and wrap it up. So in the in the gene therapy approaches that were utilized, that was announced a couple months ago, there are two approaches that have been applied. One which uses an adenovirus vector to introduce an adult hemoglobin that has that T87Q mutation. What this does is that it prevents the sickling from happening because of that anti-sickling effect. And as this is administered, it was shown that over a period of time, up to 30% of the hemoglobin had this mutation, and that was able to help the individual have fewer or no vaso-occlusive events or episodes. The second approach, which uses CRISPR technology, this actually interferes with the BCL11A production. In the absence of this transcription factor, the fetal hemoglobin level grows or increases. And over time, the percentage of fetal hemoglobin compared to the sickle cell hemoglobin changes in a manner. So here you see this blue is the fetal hemoglobin level going up beyond 6 months or so. And this allows the individual to lead a normal life without having to worry about low oxygen complications. So I know we're out of time, but I just wanted to wrap up and say that in this presentation, we learned how to visualize structures. We learned how to look at structures from the structure summary page, the model versus assembly. So this is what we were seeing. We learned how to locate where a particular sequence or residue or ligand or domain of interest is located. And we also learned to map various annotations from other sources onto the structure. We learned to look at interactions, measure distances, so that we can sort of develop hypotheses for how specific functions are being carried out in biology and chemistry. And we looked at various conformational changes in different conditions, oxy versus deoxy. So when it is oxy, what happens? When it is deoxy, what happens? And we also used MolStar and these various tools and options to develop hypotheses, save images, animations, and narrate molecular stories. With that, of course, I will thank you for your attention and the entire team for creating all these tools and resources so that we can do this. I will say that in the chat, I think Shamara is going to put in an exit survey link. So I invite you to please respond to that. And I can stay here for a few minutes to see if there are any remaining questions. And again, the recording will be made available, and we always are happy to hear from you. So please write to us and let us know if you have specific questions about a feature or a function that was mentioned here or that you have explored in the PDB. Oh, so your question was about the alignment of disordered regions. Is that what your question was? Oh, how do you zoom in on the sequence alignment? Oh, that's actually quite easy in, so if you are back, let me share screen one more time. So if you are looking at this particular display, I can actually click on, so if I scroll, I can see the sequences, and then I can say, oh, I want to look at this particular residue. And if I click on the sequence panel, it'll jump in there. And notice the same options of selection, etc., are highlighted in the MolStar window. Oh, the density surface. So, Sharmishta, do you care to elaborate? Is it how to display the density or? Okay, say I display the structure of 2DN1, and I know that this one has density information or experimental data available. So I can go to the density option here, click on enable, and see all the maps have appeared here now. If I click on any part of the structure, it'll bring up the density. And this density, I can change the representation from this solid view to the wireframe and so on. And these sliders can show me more or less. So that's changing the signal. Does that help? Okay. So I see Lakshmi Ganeshan is asking about lipid structures. Lipids are also displayed using MolStar. So any structure you can. So so I see in the chat one more question. This is probably going to be my last question about animation. So animation is something that is possible if I, let's say I hide this. And there are options over here to create animations. So I can define a trajectory of spinning, rocking, whatever. And so let's say I want to do a camera spin and whatever. And then, there are options available, and I render it and then save it. It'll create an MP4 file, and that is what will be available for just viewing as is, or you can incorporate it into a video or something. Did that answer your question, Celerino? Okay. Thank you.